Welcome to channel 360. Today our topic is forensic DNA analysis. Let's start the topic. Forensic DNA analysis, decoding genetic evidence. Forensic DNA analysis has revolutionized criminal investigations and brought justice to countless victims. Forensic DNA analysis is a cornerstone of modern criminal investigations, offering unparalleled precision in linking individuals to biological evidence. By examining unique genetic markers, forensic scientists can identify suspects, exonerate the innocent, and solve cold cases that once seemed unsolvable. Below is an overview of how this technology works, its applications, challenges, and ethical considerations. Let's see what is forensic DNA analysis. Its definition is: forensic DNA analysis has revolutionized criminal investigations and brought justice to countless victims. And its key uses are that it helps connect suspects and victims to crime scenes, solve cold cases, and exonerate the wrongly convicted. DNA analysis is a scientific process used to examine and interpret genetic material, typically by identifying unique patterns within an individual's DNA sequence. In forensic science, it involves isolating specific regions of DNA, such as short tandem repeats, STRs, to create a genetic profile that is statistically unique to each person. This profile can link biological evidence, example blood, saliva, or skin cells, from crime scenes to suspects, exclude innocent individuals, or identify victims in mass disasters. Key uses include solving violent crimes, overturning wrongful convictions through post-conviction testing, reuniting unidentified remains with families, and resolving paternity or kinship disputes. By comparing profiles against databases like CODIS, Combined DNA Index System, law enforcement can connect cases across jurisdictions. Its precision and reliability have made DNA analysis indispensable to modern justice systems, balancing investigative power with the need for ethical safeguards to protect privacy and prevent misuse. Key applications. First is suspect identification. Second is victim identification. Third is paternity/kinship testing. Fourth is wildlife forensics. DNA analysis serves critical roles in criminal justice and beyond. Suspect identification matches crime scene DNA, example blood, hair, to individuals via databases like CODIS, linking perpetrators to offenses. Victim identification uses genetic profiles to recognize remains in disasters or cold cases by comparing DNA with relatives. Paternity or kinship testing resolves familial relationships for legal disputes, inheritance, or immigration. Wildlife forensics combats poaching and trafficking by analyzing DNA to identify species, trace origins, example ivory, bushmeat, and prosecute illegal trade. These applications highlight DNA's versatility in delivering justice, reuniting families, and protecting biodiversity through precise, science-driven solutions. Now we discuss about the DNA analysis process. First step is sample collection. Forensic scientists collect biological samples like blood, saliva, hair, or skin cells. Second is extraction. DNA is isolated from the collected cells. Third is PCR amplification. Specific regions of DNA, like short tandem repeats, STRs, are copied. Fourth is STR analysis. The unique patterns of STRs at 20+ loci are compared for identification. Fifth is interpretation. DNA profiles are matched to databases like CODIS for identification. DNA analysis in forensic investigations follows a systematic, step-by-step process to ensure accuracy and reliability. It begins with sample collection, where biological materials like blood, saliva, hair, or skin cells are gathered from crime scenes, suspects, or victims using sterile tools to minimize contamination. Even trace or degraded samples may be viable due to advancements in sensitivity. Next, extraction isolates DNA from the collected cells using chemical or enzymatic methods, separating genetic material from proteins and other cellular components. This purified DNA then undergoes PCR amplification, where the polymerase chain reaction, PCR, targets and copies specific regions, such as short tandem repeats, STRs, non-coding DNA sequences with high variability between individuals. Amplifying these regions generates sufficient DNA for analysis, even from minute samples. STR analysis follows, using capillary electrophoresis to separate the amplified DNA fragments by size. Fluorescent tags attached to the STRs produce a unique electropherogram peak pattern at 20+ standardized loci, creating a genetic profile statistically unique to each individual, except identical twins. Finally, interpretation compares this profile against databases like CODIS, Combined DNA Index System, to identify matches between crime scene evidence, suspects, or unsolved cases. Matches are validated through statistical calculations to determine the probability of a random match, ensuring the result's forensic significance. This meticulous process balances cutting-edge science with rigorous quality control to uphold its role as a gold standard in criminal justice. Now move to CODIS and DNA databases. CODIS is the FBI's Combined DNA Index System, is a national database containing DNA profiles from convicted offenders, crime scenes, and missing persons. Privacy debates. Ethical concerns surround the storage, access, and potential misuse of genetic data. CODIS, Combined DNA Index System, managed by the FBI, is a national DNA database storing profiles from convicted offenders, crime scenes, and unidentified remains to aid criminal investigations. It enables cross-jurisdictional matching, linking suspects to crimes or connecting unrelated cases through shared genetic evidence. While CODIS has revolutionized law enforcement solving cold cases and exonerating the innocent, it raises ethical concerns. Critics argue that storing genetic data risks privacy breaches, misuse, example genetic surveillance, and potential bias due to overrepresentation of marginalized communities in offender databases. Familial searching, which identifies suspects through partial matches to relatives, further sparks debate over informed consent and genetic privacy. Legislation struggles to balance public safety with individual rights, as expanding databases amplify fears of DNA dragnets and misuse by authorities or third parties. Next topic is cold cases and exonerations. First, we see that post-conviction DNA testing has overturned hundreds of wrongful convictions. Second, genetic genealogy, using online databases like GEDmatch, has helped solve decades-old cold cases. Post-conviction DNA testing has been transformative in correcting miscarriages of justice, overturning hundreds of wrongful convictions by re-examining biological evidence such as blood, semen, or hair that was previously untested or analyzed with outdated methods. Organizations like the Innocence Project have spearheaded these efforts, proving innocence in cases where individuals were wrongly imprisoned, sometimes for decades. Meanwhile, genetic genealogy has revolutionized cold case investigations by combining DNA analysis with public genealogy databases like GEDmatch. By uploading crime scene DNA profiles, investigators identify distant relatives of unknown suspects, constructing family trees to narrow down potential perpetrators. This technique famously solved the Golden State Killer case in 2018, cracking mysteries that traditional methods could not. However, both advancements spark ethical debates. Post-conviction testing faces hurdles like degraded evidence or resistance from legal systems, while genetic genealogy raises privacy concerns, as individuals' ancestry data often submitted for personal use is leveraged without explicit consent. Critics argue this risks creating expansive genetic surveillance networks, highlighting tensions between justice and civil liberties. Together, these innovations underscore DNA technology's power to rectify past errors and solve intractable cases, yet they demand careful regulation to balance accountability with ethical responsibility. Now let's have a look about challenges and limitations. Contamination. Degraded or mixed samples can hinder accurate analysis. Partial profiles. Limited amounts of DNA, touch DNA, can be challenging to analyze. Backlogs. Delays in processing evidence can hamper timely investigations. Challenges and limitations in DNA analysis. DNA analysis, while transformative in forensic science, faces significant hurdles that can compromise its efficacy. Contamination occurs when foreign DNA, example from investigators, lab equipment, or environmental sources, infiltrates a sample, risking false matches or skewed results. Strict protocols, such as sterile environments and protective gear, are critical to minimize this risk. Partial profiles arise from degraded, insufficient, or mixed DNA, yielding incomplete genetic data at key loci, example STR markers. These profiles complicate interpretation, reducing statistical confidence and increasing the likelihood of ambiguous or inconclusive matches. Advanced probabilistic genotyping software may aid analysis but cannot fully resolve uncertainties. Backlogs, driven by high caseloads, limited resources, and labor-intensive processes, delay testing sometimes for months or years, impeding investigations, prolonging wrongful incarcerations, or allowing perpetrators to evade justice. Automation, funding boosts, and rapid DNA technologies aim to alleviate bottlenecks but face scalability challenges. Together, these issues underscore the balance required between DNA's unparalleled precision and the practical, ethical, and logistical constraints shaping its real-world application. Now take a quick look into historical case studies. Our first case is known as Colin Pitchfork, 1986. The first conviction using DNA evidence, proving the technology's groundbreaking impact. Colin Pitchfork, 1986. The first DNA conviction. The 1986 case of Colin Pitchfork marked a watershed moment in forensic science as the world's first criminal conviction using DNA evidence. Pitchfork, a British baker, raped and murdered two teenage girls, Linda Mann, 1983, and Dawn Ashworth, 1986, in Leicestershire, England. Despite a suspect's false confession, traditional investigative methods stalled. Geneticist Alec Jeffreys, pioneering DNA fingerprinting, analyzed semen from both crime scenes, proving the same perpetrator committed both murders and exonerating the innocent suspect. Police then launched a mass DNA screening of over 5,000 local men. Pitchfork evaded testing by persuading a co-worker to submit a sample on his behalf, but the deception was uncovered, leading to his arrest in 1987. His DNA profile matched the crime scene evidence, securing his 1988 conviction and life sentence. This case showcased DNA's dual power, exonerating the innocent, Richard Buckland, the falsely accused, and convicting the guilty. It validated DNA profiling as a revolutionary forensic tool, spurring global adoption in criminal justice. Pitchfork's capture also highlighted DNA's ability to solve serial crimes and overcome investigative dead ends, laying the groundwork for modern DNA databases like CODIS. The case remains a landmark, illustrating how genetic evidence transformed forensic science from reliance on eyewitness accounts to irrefutable biological proof. And now the second case is famous as O.J. Simpson, 1995. A very high-profile case highlighting both the reliability of DNA evidence and the complexities of legal interpretation. O.J. Simpson trial, 1995. DNA evidence on trial. The O.J. Simpson murder trial became a defining moment for forensic DNA analysis, showcasing both its potential and pitfalls in a high-stakes legal arena. Simpson, a former NFL star, was accused of killing his ex-wife, Nicole Brown Simpson, and her friend, Ronald Goldman. The prosecution presented DNA evidence linking Simpson to the crime scene, including blood matches at the murder site, his vehicle, and his home, analyzed via PCR and STR methods, then emerging technologies. However, the defense, led by the Dream Team, aggressively challenged the evidence's integrity. They argued that sloppy handling by the LAPD, such as improper collection, potential contamination, and delayed testing, compromised reliability. The infamous glove demonstration, where Simpson struggled to fit a blood-stained glove, further sowed doubt, overshadowing DNA's scientific authority. The case underscored a critical lesson: technical reliability does not guarantee legal persuasiveness. Jurors, influenced by the defense's narrative of systemic bias and incompetence, acquitted Simpson despite strong genetic evidence. This highlighted gaps between forensic science and public understanding, later termed the CSI effect, where jurors expect flawless, cinematic forensic proof. The trial also exposed racial tensions and media sensationalism's role in shaping outcomes. While Simpson was later found liable in a civil trial, the criminal acquittal revealed how legal strategy, societal context, and human error can eclipse even robust scientific data. The O.J. case remains a cautionary tale, emphasizing the need for meticulous protocols, transparent communication, and education to bridge science and justice. Now let's see our technological advancements. Till today. Next-gen sequencing, also known or called as NGS. NGS can analyze degraded DNA, expanding the scope of forensic analysis. Next-generation sequencing, NGS, transforms forensic DNA analysis by decoding degraded or fragmented samples traditional methods cannot process. Unlike PCR-based techniques requiring intact DNA, NGS sequences tiny, damaged fragments, piecing them digitally. This enables testing of aged, burnt, or environmentally exposed evidence, example skeletal remains, rootless hair. NGS also detects single nucleotide polymorphisms, SNPs, and mitochondrial DNA, revealing ancestry, physical traits, or familial links. Its precision enhances identification in complex mixtures or compromised samples, solving previously intractable cold cases and improving accuracy in victim/suspect profiling. Next are rapid DNA devices. These devices can provide DNA results on-site within 90 minutes, accelerating investigations. Rapid DNA devices are portable systems that automate DNA analysis, delivering profiles in 90 minutes at crime scenes, borders, or disaster zones. They bypass traditional lab workflows by integrating extraction, PCR amplification, and STR analysis into a single, user-friendly unit, example and rapid hit. Ideal for fresh, single-source samples, they accelerate suspect identification, verify familial relationships, example immigration cases, or aid victim identification in mass casualties. While limited for complex mixtures or degraded DNA, their speed reduces backlogs and enables real-time decisions, revolutionizing field forensics. Third is ethical AI, which can be defined as algorithms are being developed to interpret mixed sample DNA profiles, enhancing accuracy and efficiency. Ethical AI in DNA analysis involves developing algorithms to interpret complex mixed DNA samples while addressing biases, transparency, and fairness. These AI tools use probabilistic genotyping to untangle contributors in mixtures, example sexual assault cases, improving accuracy over manual methods. However, ethical risks arise. Biased training data may skew results for underrepresented groups, opaque black box models hinder courtroom accountability, and over-reliance on automation risks human oversight. Safeguards diverse data sets, explainable AI, and strict privacy protocols are vital to prevent misuse, protect civil liberties, and uphold justice. Now move to conclusion and future trends. Forensic DNA analysis has become the gold standard for identification in criminal investigations. The future holds promise for advancements like expanded DNA databases, CRISPR-based tools, and international collaboration, further strengthening this vital tool. Conclusion and future trends. Forensic DNA analysis has revolutionized criminal justice, serving as the gold standard for identification by precisely linking biological evidence to individuals. Future advancements aim to enhance its capabilities. Expanded DNA databases may integrate global profiles to solve transnational crimes via systems like Interpol's, though privacy debates persist. CRISPR-based tools could refine analysis of degraded samples or target specific genetic markers, while next-generation sequencing, NGS, unlocks detailed ancestry and physical trait data. International collaboration will streamline cross-border data sharing but require harmonized legal frameworks to address privacy disparities. Innovations like rapid DNA devices and ethical AI promise faster, accurate results, yet demand robust safeguards, transparent protocols, stringent privacy measures, and bias mitigation to prevent misuse. By balancing cutting-edge innovation with ethical accountability, forensic DNA will solidify its role as a cornerstone of justice, ensuring both efficacy and respect for civil liberties in an evolving technological landscape. That's all for today. Hit like, comment below and share this video to your friends and family. Please do subscribe if you like to watch more videos like this. Thanks for watching. Goodbye.